Dr. Adriana Corrales is lead field research scientist and underground explorers program director at the Society for the Protection of Underground Networks. The acronym is SPUN, SPUN, which is a nonprofit organization focused on the conservation of mycorrhizal fungi. Most of her work focuses on the ecology of tropical mycorrhizal associations in lowland and montane forests. Before joining SPUN, she was an assistant professor at Universidad del Rosario in Colombia for 6 years. Dr. Corrales is a forest engineer with an MS in environmental conservation from the Universidad Nacional de Colombia and a PhD in plant biology from the University of Illinois at Urbana-Champaign. She also worked as a Ewel Postdoctoral Fellow at the University of Florida. Again, thank you very much for joining us.
Thank you so much, Judy, for the introduction and for this invitation. I'm really excited to be here to tell you about the plant's best friends, mycorrhizal fungi. And I heard you talking about native plants, invasive plants, and we're going to talk a little bit about that, but underground. And I'm going to talk about what are mycorrhizal fungi and what is important for conservation, and also a little bit of restoration. So let me tell you a little bit more what SPUN is, because I work for a nonprofit that is based in the US, and our main objective is to protect mycorrhizal fungi globally. So what we do is that we produce special products like maps to understand mycorrhizal fungi around the world, their diversity, who is there, what are the knowledge gaps, and to try to cover those knowledge gaps. For example, we developed this especially model based on machine learning to predict the mycorrhizal hotspots all around the world that we just published a couple of weeks ago in Nature. So we're really excited about this. We were able to identify the most diverse places on Earth. And the cool thing, this comes with this interactive underground atlas tool that you all can access in SPUN.earth if you are curious what is the mycorrhizal diversity in your area, for example. I think you're in a very high diversity area, by the way. And these maps, we use them to protect, to basically give them as tools to policymakers, to decision makers, so they can have good tools to select the areas that we want to protect and to understand why we need to protect them. And we also want to use fungi, mycorrhizal fungi, as solutions, natural solutions for climate change. So let's talk a little bit about mycorrhizal fungi in general. So plants have interacted with mycorrhizal fungi for a very long time, and we believe fungi associated to plants were the ones that allowed plants to colonize terrestrial environments, as mycorrhizal fungi have been reported in the rhizomes, for example, of Mercantia, which is like superbasal plant species. And actually, we know also, or we suspect, that plants knew how to interact with fungi even before they had roots. And they were basically very dependent on each other. So plants have a very close relationship with fungi, and actually the root microbiome of plants has tons of different fungi on it. So we have a microbiome in our gut that helps us absorb nutrients from the food, right? Without this bacteria, we wouldn't be able to digest our food. It's very similar in the root plants because these fungi really help plants absorb nutrients from the soil. And we have different players in these roots. We have mycorrhizal fungi, which are very beneficial, right? Are mutualistic because they receive carbon from the plant and they give phosphorus, nitrogen, water to the plant. We have root endophytes as well that are much less studied, but there are also symbiotic and mutualistic fungi that live in the roots of the plants. But we also have plant pathogens. And we also have other things that always show up in these samples that we don't really know what they are doing there. There are saprotrophs, there are commensal endophytes that they are there, but we don't know what they are doing. We have fungal parasites. So there is a lot of diversity of fungi in the roots of the plants. But let's focus on mycorrhizal fungi. So basically, a lot of people have a basic understanding of what mycorrhizal fungi are because we all hopefully have seen this picture of the big plant with mycorrhizal fungi that grew better and the small plant with mycorrhizal fungi that grew less, and that mycorrhizal fungi had very good benefits for plants, right? Like they help them with nutrient acquisition. Plants with mycorrhizal fungi have higher growth rates. They absorb better, like higher amounts of water during drought periods, for example. They have pathogen defense. And even there are pollination effects through third-party interactions. So this is what we have been told about mycorrhizal fungi, but I'm gonna dig a little bit deeper on this interaction, and maybe I'm gonna give you some ecological tools that will help you understand a little bit better this relationship and understand it a little bit better. So there are several types of mycorrhizal fungi, like 4 or 5 types. The most important ones are arbuscular mycorrhizal fungi, ectomycorrhizal fungi, but we also have orchid mycorrhizal fungi. We have ericoid mycorrhizal fungi and others, a couple others that are less studied. But arbuscular mycorrhizal fungi form associations with about 80% of all the plants in the world. Ectomycorrhizal fungi form associations only with 2 or 3% of the plants, but these plants are big plants that are widespread, like conifers. Many conifers, like pines, have ectomycorrhizal fungi. And arbuscular and ectomycorrhizal fungi, they are very different types of fungi. We're talking about, for example, comparing elephants with, I don't know, snails. They differ in their biology, their ecology, and their functional traits. So more or less, arbuscular mycorrhizal fungi are very ancient, very old evolutionarily, have very few species. We believe there are like 300, 500 around the world, and they specialize in acquiring phosphorus. While ectomycorrhizal fungi are more recent in the evolution, have much more species. 25,000 is like a lower number that I would expect, and they specialize in nitrogen. So these are like, it has been shown, for example, that the ecosystem level, forests or ecosystems that are dominated by plants that form arbuscular mycorrhizal fungi, like, I don't know, ashes or ash or maples, acer, for example, have higher nitrogen content, and they have higher, basically, mineral nutrients in the soil, while ectomycorrhizal fungi that are dominated by, for example, pines, have very small mineralization rates in the soil, have very low inorganic nitrogen content, but have very high organic carbon in the soil. So basically, there is a fundamental difference in the way these ecosystems function in terms of nutrients, for example. And this is interesting because this also changes spatially around the world. We have a lot of ectomycorrhizal fungi, or dominated ecosystems in the northern hemisphere and the boreal forest, temperate forest. And we have a lot of arbuscular mycorrhizal dominated ecosystems in the tropics, but of course, the temperate forest, we have, for example, prairie ecosystems, savanna ecosystems. Those are all dominated by arbuscular mycorrhizal fungi. And those have also different environmental conditions that they exist on. So ectomycorrhizal fungi grow on or dominated ecosystems. They like lower temperatures, lower precipitations, and the opposite for arbuscular mycorrhizal systems. So you might be asking yourself, so how do I know these things for my system, right? Because we want to understand what is happening to your plants in your system. So the first, and how to basically apply the benefits of mycorrhizal fungi in your system. So the first recommendation I always make is get to know your system. What are the mycorrhizal fungi or the mycorrhizal types present in your system? So you might have ectomycorrhizal dominated ecosystems or arbuscular mycorrhizal dominated ecosystems. And that will tell you a little bit of how this ecosystem functions, right? It could have more organic carbon, it could have less nitrogen, etc. So how do you know that in real life? So there is this paper called Fungal Roots, Global Online Database for Mycorrhizal Associations. If you go look for this paper, look for the supporting information, you'll find an Excel file at the end. Then you can find the genus of your plant, and it will tell you what is the type of mycorrhizal fungi that mostly associate with your plant. Of course, they don't have all the plants, but they have many, many genera that they have studied and compiled information from. And then that could be your starting point for understanding your system a little better. So let's go back to these mycorrhizal fungi, right? They have very good benefits for plants. And then you might be asking yourself, so mycorrhizal inoculation is good for plants, right? Because mycorrhizal fungi bring all these benefits. So I want to do it in my garden, right? Is it good or bad? And the answer is not super straightforward. Our answer is it depends. It depends of your system and the needs. And we need to start understand that there is a complex interaction between the plants, the microbiome in the soil, and the biotic conditions of your site. So everything is context dependent in ecology, basically. So on one side, you have the plant identity. Who is your plant? What are the characteristics of your plant? On the other side, you have your fungal identity. Who is your fungi? And what is the characteristic of your fungi? And we know that there are multiple plants and there are multiple fungi interacting at the same time. Also, these plants and fungi are interacting with other organisms like herbivores, pathogens, bacteria, etc. And they are interacting with the environment as well. So we have different soil types with different nutrient conditions. Their experiences different light conditions, different water conditions. And all this is gonna affect the interaction between that plant and that mycorrhizal fungi. So I'm gonna give you some elements that are gonna help you understand a little bit more what is going on. So for plants, you're plant people, so maybe you already know this. We have different types of plants depending on their functional diversity or the type of ecosystem they can survive on. So we have plants that are stress tolerant, that are able to grow in very stressful environments, in very dry soils, in saline soils, or under permafrost, something like that. They can tolerate some stressful conditions. We also have the rural communities that are the first plants that invade an ecosystem after a disturbance. And we have also the competitive ones that are able to survive in places that are already full of other plants and where nutrients are not very common. And all those things are represented by different traits in these plants they grow faster. For example, the rurals, they go faster. They could, they reproduce faster, they produce a lot of seeds, they are dispersed by the air, things like that. The same thing, and this is the reason why when you are selecting your plants for your garden, for your space, you are not just gonna go to the store and grab whatever seeds there, right? Because you are thinking, what are the plants I need to put in my soil, this type of soil and these conditions to get these results, right? And the same happened with mycorrhizal fungi. Mycorrhizal fungi also have different functional groups. There are mycorrhizal fungi that are adapted to very disturbed ecosystems, while there are other mycorrhizal fungi that are stress tolerant, and there are others that are competitors. So not all mycorrhizal fungi could be applied to every environment. And as we select seeds, based on their characteristics, we need to select fungi or mycorrhizal fungi that are good for the conditions we were looking for, we have in our garden, for example. And also, we have to look at the traits of the plants, right? The characteristics of the plants, the characteristics of the fungi, and the characteristics of their interaction, because not all fungi are gonna want to associate with all plants. And not all fungi can survive in all ecosystems. So we need to really look at the three aspects of these, which are the plant, the fungi, and the symbiotic traits. That's when they get in contact with each other. And of course, we need to look at the morphological traits, at the physiological traits, and phenological traits. But regarding of all this complexity, these fungi, mycorrhizal fungi, are not very well studied and not very well represented, for example, in restoration management plans, or basically in any type of management plans, because they have been quite understudied. And also, there is a huge bias to above ground, right? We get super interested in what we can see, what we can touch, but things that happen under our feet are not super visible, and we are not aware of it. And scientists have the same bias. We like to see and study what is easy to access, what is easy to see, but when you really need to use a microscope, dig, or it just gets too complicated, and we usually don't get to study it. So yeah, mycorrhizal fungi are not very well represented in these plants, and that's of course a mistake, because, for example, it has been shown that the use of these native mycorrhizal fungi are particularly important for native late successional or plants with conservation concerns. So if you have plants that are slow growth, they only are found in pristine ecosystems, or also that are so rare that they are disappearing, there are high probabilities that they associate with some type of fungi that are super special and that are also associated to these late successional stages in the ecosystems, or fungi that are also rare and also potentially endangered. So, for example, in this paper from Tanya Chiki from 2019, she shows exactly that plants that are late successional are more responsive to inoculation with native mycorrhizal fungi. So basically, if we want to preserve plants and also plants that are endangered, we really need to think about their microbiome and how we are going to conserve these plants with all their biotic interactions, because plants cannot live by themselves. They need their mycorrhizal fungi, they need their pollinators, they need all those organisms that they interact with to be able to survive in the future, right? So this is super important because yeah, this microbiome is really, really interesting and important for conservation and restoration. So there are two types of inoculum that is used for plants. We could use live soil, basically a scoop of soil that we took from the forest for a very well-preserved area and use it as source for our mycorrhizal fungi, or we can also get inoculum that are mycorrhizal fungi that has been isolated and has been grown and are offered either as inoculums or as an inoculum, as commercial inoculum or non-commercial inoculum. And for example, in this study, they also show that plants are very responsive to inoculation with these types of inoculum. One important ecological concept that I think is key to understand the interaction between plants and their mycorrhizal fungi is the priority effect. So when plants are in contact, or a seed is starting to germinate and gets in contact with the soil, with the inoculum, then it has been shown that the very first species that colonize that root has an advantage. For example, this experiment from Peter and Kennedy from 2009 with ectomycorrhizal fungi, they show that the very first species that colonize is the one that is going to dominate in the community with several species. And that has been also shown for arbuscular mycorrhizal fungi, where plants basically they have a limited amount of space to host fungi in the roots. And basically the initial colonizer is the one that in many cases basically wins all the space and is the one that is gonna have a relationship with the plant for a long time. And this is so important because sometimes we think that we want to grow seedling, we can grow seeds on something, some temporary soil, and then we can transplant them later when they are going to find their mycorrhizal fungi. But if they are not paired with something that is really good and very positive from the beginning, chances are that later on they might not be able to get their right symbiont. So that's an interesting. So let's jump to this other topic that is a little bit of hot topic and in mycorrhizal use, and is the type of the inoculum that we use. What is best, commercial inoculum or live soil inoculum? And I'm gonna tell you a little bit what is this about. So what do I know about commercial inoculum? You probably have seen commercial inoculum being sold in stores. There are many products, and unfortunately, what it has been shown when this inoculum has been tested is that a lot of the inoculum doesn't really form effective colonization when it's applied to the plants in the soil, either in pots or in the field, because first, apparently it has low propagable viability. It's probably a lot of exotic species as well, or species, mycorrhizal species that are not adapted to your local conditions or are not adapted to your native plants that you're trying to grow. And also, super concerning, they could have pathogen contamination. So instead of inoculating your plants with very beneficial mycorrhizal fungi, you're even sometimes inoculating some pathogens. So it has been shown by Liz Koziol in this 2025 paper that inoculating with field soil or with high or like lab-grown fungi, which is basically the inoculum that she was producing was much better than inoculating with commercial products. And also, they also found that these products don't have as many propagals as they claim they have. And also, when they inoculated the plants with the commercial products, some commercial products even produce negative. That means negative results. That means the plants grew less when they were inoculated than when they were not inoculated with any type of product, which is like super concerning. And of course, this is not a black and white topic, because as I had been talking about, there are many intermingling factors that could affect this interaction. But in general, commercial inoculum could have some dangers. For example, the introduction of exotic invasive species, like, for example, Amanita muscaria or Amanita phalloides, right? The caps are invasive globally. They are mycorrhizal fungi that were introduced, for example, in the case of Amanita muscaria, introduced in many areas in the world to grow commercial plantations of pines, right, for timber production, and is a huge invasive species. And this could be and this is very visually striking fungi that is, of course, we can see it, but for arbuscular mycorrhizal fungi, they're mostly microscopic. How are we gonna know we are introducing an exotic species that is becoming invasive? Is very hard to monitor, for example, these changes in underground ecosystems. So we need to be extra careful with the type of products that we use. In this paper from 2006, for example, Schwartz et al. Look at different characteristics of the fungi that are used in inoculum to try to avoid these potential problems. For example, we want to look at host compatibility. If a fungi has a very broad host compatibility, that means can associate with any type of plant, it's very easy that this species is going to escape cultivation and is going to go into the wild and become invasive. Dispersal, right? We don't want airborne spores being dispersed everywhere. And these are very similar characteristics to invasive plants, right? That are very competitive. Species that are very competitive could be very competitive outside of their native range and become exotic, invasive. When there is a very fast growth rate, same. Or in fungi, when they have saprotrophic capabilities, in addition to mycorrhizal capabilities, they also could escape and become saprotrophs or become pathogenic for other species. So all these traits are important to look, and they will cause impacts on the local mycorrhizal communities because they are going to start competing with the locals. They have impacts on the local plant species, and they will have impacts on the local ecosystems. Because this fungi could really change, for example, nutrient cycling. So we need to be careful with the purity of the inoculum, for example. We have to be careful that we're not facilitating plant invasion, for example, co-inoculating things. And also, as I said, we need to be careful that they are not taking a lot of energy from the plant or carbon from the plant to reproduce. So these authors have three very compelling, I think, advice for when you are selecting inoculum. First, really think about, do you really need to inoculate mycorrhizal fungi? This is super important because, as they say, mycorrhizal fungi are not always beneficial because of these complex interactions, but also because mycorrhizal fungi could be available in your soil, but you really haven't realized. So it's important to assess this. Also, if possible, always use local inoculum sources to avoid invasive species for introduction, for example. And also, they recommend use sterile produced cultures instead of live soil, because also you could be introducing pathogens to your system. So basically, we want to look at the germination, the colonization, the persistence, and the spread of this fungi to be able to select what is best for our plants. So Miranda Hart, in this functional ecology paper, had a very nice and useful, basically step-by-step decision chart where you can really think, do I need to inoculate with arbuscular mycorrhizal? I don't know. But do I have mycorrhizal fungi in my soil that I already have and I don't know? Or yes? And then step by step, you can start making a decision about if you want to inoculate or not. And basically, in many cases, you only need to inoculate when you basically lost all your topsoil and the soil is completely degraded. In most cases, you don't need to inoculate with foreign species, for example, or with just external species, and you can just rebuild your local communities from natural sources nearby. And also, it's very important to have very careful inoculum selection, careful application, and also know what is going to happen after you inoculate. Because, as I said, these species can escape, and we still lack information about what happens to mycorrhizal inoculum after we applied it to our plants. This is something at the ecosystem level, this is something that we're still trying to figure out, and we don't know exactly what happens after these species are established and they start breeding with the local strains. We don't really know. Okay, this is about inoculum, and now I'm going to talk about live soil inoculum, which is, as I said, taking a scoop of the soil and just putting around your plants to try to make the plants grow better. In this case, this is very interesting because live soil, we know like soils are just mega diverse, and there are not only mycorrhizal fungi there, but there are tons of bacteria, microarthropods, viruses, etc. that are there. And in this paper by Avril in 2022, they argue that inoculating with live inoculum, live soil inoculum, help restoration plants, basically plants that were producing for restoration purposes, grow on average 64% more than other plants. That plants that were not inoculated with live soil inoculum. So that's striking, right? Because of course you are not only inoculating mycorrhizae, because there are also complex interactions with bacteria that help plants grow. And yeah, live soils could be very beneficial. Indeed, live soil inoculation has been proposed as innovation for assisted forest restoration. One of the main issues is, where are we going to get so much live soil to inoculate all these restoration areas that we want to restore, right? So we need to really find a way to produce this inoculum without using enormous amounts of soil for it, and because it has to be healthy soil. It cannot be any type of soil. We want to use pristine, well-preserved, healthy microbiome for these purposes, right? And that's a very valuable resource. So it has been tested. It's been it has shown very good results in extremely challenging habitats, for example, with extreme conditions, and also when habitats has been basically the soil has been very, very affected. And this has been shown, for example, that the mycorrhizal community present in the soils is directly associated with the growth of the trees in a forest. So when you are inoculating with this live soil, you can know that the number of species and also the species composition of the soil is directly associated with the growth rate and the survival rates of the seedlings and in the long term of the forest in general. But there is another element that I think I wanted to mention, and it's pathogens. Because when you are inoculating live soil, you are inoculating everything that is there, including the pathogens. So I believe not all plants are as responsive, will respond positive or negatively to live soil, and that would depend on the characteristics of the plant. For example, usually plants that are late successional, that means that grow slow, they have low specific leaf area, have strong defenses, respond positively to inoculation with live soil, while species that are ruderal, that grow fast, reproduce a lot, and have poor defenses are going to be very susceptible to pathogens and therefore very susceptible to these live soil inoculations. So we need to be careful because it's not like one method fits all. We need to think about all these characteristics. Also, for arbuscular mycorrhizal systems that have more nutrients, we have seen also that they grow more pathogens. While ectomycorrhizal systems have less nutrients and have less pathogens. So in this case, they are going to be more responsive, are going to have a more positive response to live soil inoculum than other plants. So this doesn't work for all cases. So to summarize a little bit, fungi are not have not been historically considered in this conservation, in our conservation plans, in our restoration plans. And as we know, like plants are so important for restoration and conservation, and they completely depend on this fungi for survival. So it's important to start including these mycorrhizal fungi in our conservation plans. Also, we're restoring a symbiosis, and this is a more complex interaction that we think of. And so we need to think about what plants we are working with, what are the mycorrhizal fungi they're associated with, and what are the abiotic environment that they are going to be interacting, because all that complexity is what's going to actually tell us what the outcome is. Commercial inoculum is not always the best. It's only recommended when there is not other options, but it's better to use local sources. And also, in general, it's like low quality. Natural inoculum, like live soil, could be very beneficial, but I don't think this is gonna be also applicable in every situation. So we also need to understand the complexity and try to decide if this is the best option for us or not. So I'm going to show you a couple of, in the little time that is left, a couple of study cases that I have done in tropical forests here in Colombia, but these are widely applicable for many systems. And the first, quickly, I want to show you how mycorrhizal fungi, for example, could differ between natural and urban ecosystems. Because if we are thinking about sourcing inoculum from nature or from more natural, pristine ecosystems, we need to understand what are the differences. So with one of my students, when I was at University of Rosario in Bogota, we basically collected the roots of oaks growing in the city and growing in a natural preserve near the city. And we found that there was a huge difference in the community composition of this mycorrhizal fungi. So this on the left is the natural site, and in the right are all the urban sites. And as you can see, it's just super different. We found that in the natural sites, we have Russula and Lactarius that are not associated with high nitrogen or extreme conditions, but the trees growing in the city associated mostly with scleroderma, which is a fungi that is associated to harsh environments, especially very dry environments, and also with Laccaria, that is a fungi that is nitrophilic, that likes high nitrogen conditions. So if I'm gonna, for example, get inoculum sources for a plant that I'm trying to grow for a restoration, I might want to try to outsource that inoculum from a natural forest instead of, for example, a more urban forest that have a completely different composition of species. Another example that I have here is with my student, Luisa Fernanda. We look at this cocoloba tree, which is the sea grape. This is an ectomycorrhizal plant, but when we talk about this project initially with the Botanical Garden of Cartagena, where they were just growing seedlings for restoration, and they didn't know this plant have a different, completely different mycorrhizal association to all the other plants that they were growing. And they were just using commercial soil on it, and they weren't growing very well. So this is in the north of Colombia. So we decided to run an experiment, growing seeds of cocoloba into initial substrates, peat and sand, sterile, and native soil, just germinating the seeds. And when those seeds germinated, we moved them to commercial soil or commercial soil with an inoculation of native soil. And basically, we followed these seedlings for 6 months. We measured their growth rate. And at the end, we sequenced what was in the roots. And we found really interesting results. Basically, what we're seeing here is the two bars in the left are the seedlings that were grown in native soil, the seeds that were germinating in native soils. And the 2 bars in the right, or the boxes in the right, are the seeds that were grown in peat initially. And as you can see, the seedlings that grew the most were the ones that were grown or germinated in native soil. And then it doesn't matter what you move them to in the next phase. What the initial microbiome with the seedlings germinated was the one that determined how much these seedlings were gonna grow, at least in these six first months of life. But when we sequenced the roots of the plants, we also found that the seedlings that were germinated in native soil and then grown in native soil were the ones that have the most ectomycorrhizal fungi in the roots. And also, interestingly, the seeds that were grown in peat initially and then transplanted to commercial soil had no fungi in the roots. So they didn't amplify. We couldn't get fungal DNA from these seedlings. And I think that's probably not very good for the seedling resilience, for example, when they are transplanted into the soil, because they are not going to have these fungal friends to help them. And the last experiment that we run was this with Juglans neotropica, which is the walnut. It's a walnut that is endemic from Colombia. And then what Mariana did was we tried 2 different treatments. We used 2 different seeds that originated in 2 different populations, and then inoculating them with native soil or commercial soil as well. But here we track every single inoculum source. So we went in the field, we collected inoculum, live soil from 3-4 different adult trees of the same species, and we track who has which inoculum. And we found two interesting results. First, that when you use native inoculum, live soil inoculum, the seedlings will grow faster, bigger than with commercial soil. And also that not all inoculum sources work the same. There was one inoculum that was just especially particularly good, and seedlings grew much better when they had this inoculum. Also, when we compare the effects of this inoculum in seedlings from the local populations versus seedlings from a different population, seedlings from the local population grew better on the local inoculum than the seedlings that were coming from a different population. So we don't know if the seedlings from the local population were better adapted to the local inoculum. That's something that we are not going to know from this small experiment. But we did find that the significant positive results were from the local seeds and not from the other population seeds. So that's something interesting to look, for example, for you that work with local native species, that it's very important to have inoculum that is local, because for some reason there could be something that is special, species that are adapted to your local soils or something else. When we sequence the seeds also, we at the end, we harvested the seedlings, not all, but some, and sequence everything that were in the roots. The seedlings that were grown in the greenhouse had a lot of pathogens also. And we think this is associated with the peat that we were using. But also, yes, the community composition of the seedlings that were grown with commercial inoculum were completely different than the ones that the native seedlings, and had less mycorrhizal colonization. Okay, and this is just, we have an associates program at SPUN. If you are interested in learning more, you can go to our website and look it up. You can join the network if you are interested. And yeah, thank you so much. That's all I wanted to share right now.
Thank you, Adriana. We have a question already. Lauren, go ahead.
Hi. Yes, that was amazing. That's very fascinating work. I have a lot of questions, but I'll try to. I think I'll pick one that I think might be really interesting to a lot of people, which is I'm thinking about, situations where a restoration project is happening and they have perhaps scoured the topsoil and removed the topsoil. Maybe they've stored it on site to put back, but it's very disturbed. And what you often end up with is a lot of invasive species, especially the ground layer. Maybe there's some tree planting going on, but the ground layer is a mess of invasives. Happens often. And I'm wondering, I couldn't quite figure out from your description whether or not the common invasive species that have high growth rate and certain, invasive tendencies, right? The way that they seed and so forth, would they be more likely to be problematic if you added mycorrhizal additions? Like what way would you go with that situation in terms of adding or not, or using the local material? Like, how do you think about that?
Yeah, that's a great question. And I think this is something that we are still figuring out, because there hadn't been a lot of research on restoration in mycorrhizal fungi. But yes, definitely what I think is this invasive are usually very rural, very weedy. And also for example, the fungi that are sold commercially in this inoculum are also the ones that love that type of plant. They're weedy, mycorrhizal fungi. So applying inoculum could actually benefit the invasives and the fast growing. Now, using the topsoil that is left after the intervention, I think that would be the best if the ecosystem was in good shape beforehand, right? Because we want to use inoculum that is as close as possible to what the plants that we want to grow need, right? So I think using the local sources of soil would be much better than using commercial inoculum. But understanding what is the condition ecologically of that ecosystem before it was disturbed will be also important to try to decide if we want to use that inoculum or not. I don't know if that makes sense.
Yes, I have a follow-on to that then. You mentioned that and I looked at your website, and actually for folks, I posted a link to it in the chat. If you look at our maps in our region, you'll also find that we have really high levels of invasives. So we begin with usually a fairly invaded landscape. Maybe the trees are still good. There's often pretty decent spring ephemerals, but there's a lot of invasives. So assessing the health of the population to begin with, like, how do you even think about that? How would you go about deciding whether or not your inoculum was good, knowing that, yeah, there's some good things underground there, and we do have some trees, but we have kind of a hellscape of woody invasives or kudzu or whatever, vines or porcelain berry. I mean, we have all number of issues in our area.
Yeah, no, that's a great question, and that's the 1 million question, because I don't have an answer for that. But I think it would be worth it to try to do some sequencing of at least a few samples and try to see what species are there and if those species are worth inoculating to the rest of the community. Because it's very important to have an inventory of the species that are present. And I know this is a little difficult for mycorrhizae because you need to use DNA methods, but I think setting up that baseline would be really important to try to make more informed decisions about your site.
Thank you. That was amazing. I'll let other people ask questions.
Lisa, you have a question?
Yes. First of all, thank you again for a wonderful talk. Just two quick questions. One is, as was just mentioned, we do have a lot of invasives here. And so a lot of times for restoration projects, we end up using herbicides to treat those first. And I'm just wondering, do we know if herbicides, I'm thinking that they would, but do we know how herbicides impact mycorrhizae? And also I was wondering, is there a place online maybe where we can purchase local inoculum?
Yeah, that's a great question. From what I have read, yes, herbicides could affect mycorrhizal communities. I think there's a little bit of research on that, but not a lot. And I don't really know in Maryland if there are local sources of like commercial inoculum. Yeah, that's a good question, because I know there are few companies that are starting to emerge producing local mycorrhizal inoculum. I know one in Kansas, but that's far away. So I don't know any locals, but yeah, I don't know if that answers your question.
Thank you.
Christina?
Great presentation, Adriana. Thought-provoking. My question is about how this inoculum approach could work on acidic soils or soils with low pH. such as the one we find in Eastern Shore. There's always association about low pH and fungi not thriving as well. So I would like to know your thoughts about the inoculum approach. It has ever been studied in low pH soils.
Yeah, no, that's a great question. And that's part of that interaction and locally adapted communities that I was talking about. For example, mycorrhizal fungi or the soils here in Colombia, our pH is between 3 and 4 in the Andes.
Oh.
And there are mycorrhizal fungi that are adapted to those environments. So when you have those extreme conditions, I think it's even more important to use your local inoculum that is adapted to those conditions and the plants that are adapted to, so they could have better interactions and you could have better results, because if you apply this commercial inoculum that was grown like in a neutral soil, for example, it's probably not gonna be able to survive and even less compete with the local communities. So that will reinforce like the importance of the local communities. Yeah.
Interesting. Thank you.
We have a question from Karen. Is leaf grow a good product because it's made locally?
Oh, is this a mycorrhizal inoculum?
No, it's not. Just
Basically, it's everybody's yard waste that gets turned into a compost that is sold back to the community. So people put out their yard waste, they take it off to a facility, compost it, sell it back as a basically compost.
Okay.
and
Oh,
it does include both native vegetative waste, but also non-native and invasive vegetative waste.
okay, okay. Yeah, that's difficult to assess. But in terms of mycorrhizae, I don't think you're gonna find a lot of mycorrhizal fungi in that product. So yeah, it seems like it could go both ways, and I have no a good answer for this, but yeah, usually mycorrhizal commercial inoculum or inoculum are made of spores that are isolated, then they are inoculated on a trap plant and then amplify through time to reproduce and have a bunch of spores. And then they sell that soil that is enriched in these spores. So I think if it's just compost, I think it's very unlikely that you're going to have a lot of mycorrhizal fungi there.
In addition, it is made in the process of making it, it actually goes to quite high temperatures.
So they kill off all the microbes, probably good and bad.
Exactly.
Okay.
More importantly, they kill off the weed seed, which is difficult.
Supposedly, they kill off the weed seeds.
Patrick asks, when rescuing plants that would otherwise be destroyed, how do you bring necessary mycorrhizal fungi with the plants? The only way that he can think of is to take a larger root ball.
Oh, that's a good question. So most plants, when they have already been in contact with the soil, they usually, if they are mycorrhizal, they usually have colonization already in their roots. So if you're rescuing a plant that is already grown in native soil or in contact, it probably has already its mycorrhizal fungi associated. And I don't think you will not need to inoculate again. I think the main problem is when you are growing from seed and that seed is going to be exposed to an environment that is not natural. Then you will need to inoculate.
Following up on that question, I think one of the things the person is asking about is how much of the soil should be brought with the plant to get enough of the mycorrhizal fungi.
Yeah, that's a great question too. When, for example, I grow sterile seedlings and then I want to inoculate them, When I'm gonna use live inoculum, for example, in these experiments that I show you, I only use 6% of inoculum, and the rest could be any type of background soil that goes well with the plant. So the important thing is that the soil is in close contact to the roots, and then they are gonna work it out. The mycorrhizae and the plant are gonna find each other. But the amount doesn't have to be a lot. And very importantly too, you want to stimulate the symbiosis, the formation of the symbiosis, you should use low fertility background soil, because when there is a lot of nutrients, many mycorrhizae don't like that, and plants tend to just try to get the nutrients on their own without forming the symbiosis. So trying to keep low fertility will make them more likely to interact.
So I'm going to go a little out of order here because Carol E. actually has a question that's kind of a complement to that last question. If you do root washing when repotting plants, does this affect the beneficial effects of mycorrhizal fungi?
Yes. Yes, I wouldn't recommend to wash roots because mycorrhizal fungi are very microscopic. They are mycelium. There are large networks that go out exploring the soil. When you pull a plant, little bits of it could be still stuck to the root, but when you wash it, you're probably gonna lose most of the mycelium, and then the fungi are gonna have to regrow all that mycelium. So ideally, you are gonna try to not disturb the plant as much as at the root as much as possible, and something that is called the rhizospheric soil, which is the few millimeters that are around the root. Those are where most of the microbial communities that are beneficial are concentrated. So yeah, I wouldn't watch it because also you will lose your bacteria. I think unless there is like a big need for it, I wouldn't recommend it.
Chris asks, when studying the microbiota of plants, is it possible to do blast DNA sequencing the way some people are able to do with the human gut microbiome? Or is the technology or resource availability not there? Is the sequencing too hard or whatever?
Oh yeah, that's a great question. Actually, that's what we do at SPUN. We work with environmental DNA of the soils and the roots. So basically, we take a piece of root or a little bit of soil, it's like less than a gram. We extract the DNA of the whole microbial community or the whole mycorrhizal community, and then we do Illumina sequencing or environmental sequencing with any platforms. And then we're able to identify all the fungal species that are present in that root or in that little bit of soil. And it's exactly the same method that it has been used to characterize the human microbiome. So there are so many similarities with this mycorrhizal ecology and root ecology and our gut, because it's basically a very similar environment and the methods have been very similar as well.
We have a couple other questions about things that might hurt mycorrhizal fungi. So one of them is, has kind of the loss of mycorrhizal fungi been made worse by builders who scrape off the local topsoil and then replace it with whatever after construction?
Oh yeah, absolutely. That's so sad because soil, not even mycorrhizae, but the whole soil takes millions of years to form, right? Soil is like not renewable, and take it out, remove it, and replace it with, I don't know, sometimes they even put plastic on it, on like the surface is a big loss. And also soils are mega diverse, so you are not only losing the mycorrhizae, but everything else that lives there. So yeah, it's that hurts a lot. That's like the worst, like the nuking the mycorrhizal communities. But also applying tons of fertilizer is bad. Even tillage is not super good for mycorrhizae because they have these networks and they depend on the network to obtain the nutrients. When you are doing tillage, you are breaking those networks as well and making them less efficient. So also those little things could affect mycorrhizal fungi as well.
Another question about what might affect the mycorrhizal fungi. Nancy asks, how does glyphosate affect the health of mycorrhiza?
That's a great question. I think glyphosate kills pretty much everything. It's pretty bad because it's like a herbicide, kills, it doesn't kill everything, but I think there's gonna be some effects. I haven't come in contact with any research studying specifically this chemical, so I cannot say anything about it. But yeah, I am not sure where it's. Yeah, I don't know, but my feeling is that it could affect the soil microbiome.
Lauren,
Cool.
you have a question?
Yeah, follow on with that, the herbicide question. So I'm thinking, it kills, glyphosate, of course, kills the plant on top, and then eventually that is going to kill the roots as well because it travels down, translocates down. But I'm wondering if you're not necessarily disturbing the soil, can those mycorrhizae survive for a while? So I'm thinking of, again, these restoration scenarios where you have a site that's just covered in invasives and really choices are all bad, honestly. It's herbicide or you till it all under repeatedly because that one time won't work, whatever. So in those scenarios where you do the herbicide treatment, can the mycorrhizae survive for a while all the plant life is dead? Where you've really just done a full kill and then you seed into that.
Yeah, no, in restoration, you just need to use trade-offs. What is less invasive, I totally understand. And it's never like a perfect solution. So mycorrhizal fungi, mostly arbuscular mycorrhizal fungi, are very highly dependent on plants, but they could create spores that could potentially survive for a while without a host, but maybe not months, but some time without a host. So I would say, yes, if everything dies, there might be some mycorrhizal that survives, but it's not ideal for the mycorrhizae. For ectomycorrhizal communities, there are more chances that they are gonna survive because they have more extensive mycelium networks, and also some of them could become saprotrophs in the absence of a host. So I would say those could survive even longer without plants around.
Again, going from that point and the trade-offs, besides tilling or herbicides, there are two other possibilities. One is solarizing by covering the weeds with plastic. And another is what people call the lasagna method, which is putting mulch and cardboard down in it, at least one layer or possibly more, but inches thick. How does the damage to the mycorrhizae compare there versus tilling or herbicides?
Oh, that's a great question. I would say that adding organic matter like in the lasagna scenario, I think it would be the least detrimental for the mycorrhizal fungi. I think because it's like there is no disturbance of the networks and there's no super high temperatures. So I would say that could be the more benign method.
Another question about mycorrhizae and invasives. Linda asks, can mycorrhizal inoculum assist with deterring invasion of areas by foreign invasives? And also, what are the local sources that assist the quality of mycorrhizae or the symbiotic relationship for the local plants? And I think you did cover a little bit of this earlier, but I think that this is a more kind of focused question about it.
Yes, yes. So what I have read from the literature and what I show you is that, yes, the late successional species that are more of the native species that we have these conservation concerns are more responsive to native mycorrhizal inoculation. And I came across some research that said that inoculating with native mycorrhizae might reduce slightly invasion from exotic species. So having a healthier mycorrhizal community could help a little bit reduce invasive species, and I believe is because invasive species in most of the time are those rural weedy species that are looking for weedy mycorrhizal fungi. And if we have more of a late successional mycorrhizal community, then they are not going to have good partners to thrive. And this could reduce a little bit. I don't know how much, and it would probably scale by case scenario, but it could help a little.
Carol asks, we have been using recently ground wood chips and newspaper and cardboard for preparing the ground ahead of planting native mini meadows. This seems to work well and fosters more rapid growth of plug plants. Are there things that we should look at more carefully, such as the type of tree that was chipped? And I'm guessing that Carol's referring here to what we're just talking about as lasagna method. But I guess her question is, anything that should be looked at when you're actually using that method in terms of making it more effective or less detrimental?
Well, that's interesting. I really don't have information about the effect of the type of organic matter that you're incorporating on mycorrhizal fungi per se. But probably I would say, yeah, I think mycorrhizal fungi will like probably more. Yeah, it's hard to tell. I don't have evidence in my head to really compare. But yeah, that's an interesting question.
Carol, a different Carol, asks, is it possible when growing perennials, can two plants side by side need different inoculants?
Yes, absolutely. Is the example of the maple and the pine, for example. Maple is arbuscular mycorrhizal, pines are ectomycorrhizal, and they will have completely different mycorrhizal types and completely different inoculum. So yes, now that doesn't mean that they are gonna affect each other. They could happily live together probably if they have their mycorrhizal fungi available. But yeah, some species might start competing, but there are a lot of mixed forests that have both, like oaks and pines, sometimes grow together happily, and it would depend on the plant. But yes, you might have to use different mycorrhizal inoculums in some cases.
So we had a couple of questions about, where to find local inoculum, and Tiger comments that the Maryland MDR has a list of native plant nurseries that carry local inoculum, and he put in a link in the chat to that.
Oh, that's wonderful. That's so cool.
We're getting towards the bottom of the questions here.
Well, I do want to say that this has been an amazing presentation with lots of very useful information. I'd like to change the topic. It's getting a little late, but I would like to ask just something that I know has been touched on in the past, which is how do mycorrhizae affect global warming?
We think that mycorrhizal fungi, this is small fungi, they're microscopic and they might not affect very much the carbon cycle, for example, at the global scale. But what we have found is that basically mycorrhizal fungi are the circulatory system of Earth because they create kilometers and kilometers of these hyphal networks that move 13 billion of carbon, 13 billions of tons of carbon every year, which is equivalent to 1/3 of all the emissions from fossil fuels. So that's a lot, right? And it's just microscopic and it's happening underground. We don't see it. We don't realize it's happening, but it's a huge carbon sink because, of course, plants take up the carbon from the atmosphere, but a lot of that they traded for nutrients with the mycorrhizal fungi. So mycorrhizal fungi access that carbon directly and put it underground where it is less likely that it's gonna decompose. So basically, I think mycorrhizal fungi could be very much a very good natural solution for example, climate change, if we use it correctly.
Thank you. And is there anything else that you would like to say in summary or something that wasn't touched on?
No, not really. I just very happy to talk about plants because I love plants. As you can see, I'm just obsessed with plants. I love plants and I love mycorrhizal fungi. And I just want you to think like, every time you think of plants, think of fungi because there are tons of fungi living in the roots, but also in the leaves. The microbiome of plants is just full of fungi and they have very complex interactions and it's just fascinating. And I really appreciate people that think about native plants. It's so important. And native fungi now, I think you're gonna start thinking more about native mycorrhizae and yeah, there is a lot of diversity. If you want to learn more, go to our website. We have explainers about how to improve mycorrhizal fungi in your garden, for example, or explainers from our new paper. You want to learn more, so just visit our website and we have more information there. Thank you so much
Again,
for having me.
well, thank you so much for taking the time to talk with us and being patient with all our questions. Just a terrific presentation and wonderful information. Thank you.
Thank you so much. It was very fun. Have a good night.